Thank you for the organizers to actually tell us a bit about what we do in NAD. So I am the co-founder and CEO of the company, but also I still work partly at the University of Helsinki. And NAD is a spin-out company that is now about 2.5 years old. And we have spin-out from the University of Helsinki from the group of Professor Anu Suomalainen-Partiovaara. And in her group, we are studying mainly the mitochondrial disorders and the metabolism. So I don't think I need to go into the detail what NADs are standing for and how important they are in the metabolism. So of course, in the studying mitochondrial disorders, we mainly use mouse models. And in 2014, we have published a study when we actually use a mouse model for mitochondrial myopathy, where we treated the mice with NR. And at the same time, in Switzerland came a similar study where they also treated mitochondrial diseases with the NR. And this is an interesting study where it shows that NR actually helps improve the muscle metabolism of those animals. And of course, the results were so interesting and important that we thought that this should be transformed also to the human clinical trials. But unfortunately, the transformation from mouse to human is not always so simple. So with the mouse, you can just take whatever tissues or cells, send it to a mass spec facility and wait for weeks and months for the results. But if you want to do an intervention in the humans, that's not really what you can do. So you want to and you need to follow up the patients. And the easiest tissue in the humans where to follow the NADs would be blood. But at that time, we actually didn't find any technology or any possibility how to, in real time, measure NADs from blood. And one colleague of mine and the current CSO of NAD, Lilia Euro, took up the challenge and started developing NAD assay. And she was very successful and developed something what we now call NAD technology. And there we start with any material, blood or tissues. We do one-step extraction where we get all the extracts and with the extract we can then divide into different parts. We treat them and then we analyze them individually, each metabolite in a single plate. And this whole thing can be, or the whole analysis can be done in any research lab using regular equipment, so you don't need to use any sophisticated equipment, and the results are equal to, with the accuracy to mass spec. So using this technology, then we were able to proceed with a clinical trial where we had 6 mitochondrial myopathy patients, and we already at the beginning established that their NAD levels in the blood was definitely lower than in the controls. And after 6 weeks of intervention with niacin, their NAD levels increased and stayed high throughout the time. Also, in this paper, we also showed that the muscle NAD levels increased and the patients did really, really well afterwards. One question then, of course, afterwards, many papers studying NAD metabolism in humans and mice and clinical trials then appeared. But one thing that stayed kind of still open was what is actually the normal range, what we are comparing our patients to. And so with using the NAD technology, we were then able to establish what is now called the normal reference range based on 300 healthy individuals. And this is in the whole blood range of NAD+ and NADH. We still were then interested in the main question, which would be, what are the effects of actually healthy people taking the NAD boosters like niacin, nicotinamide riboside, or NMN? And that's why we took the controls from our original clinical trial that had niacin for 16 weeks. There was an increased dose from 250 milligrams per day to 1,000 milligrams, and we, of course, sampled them throughout the time. And we focused purely now on the healthy individuals. And here you can see that we can monitor with NAD technology the increase in NADs in the blood. Here's the green is the normal range that we have established. And you can see that the levels are nicely increasing throughout the time. But also what is interesting is that both of these metabolites then reach a saturation level, which is for NAD+, it's between 85 and 110 micromolars in blood. And then for NADH, it's about 2.5 micromolars. So the NAD levels do not increase endlessly, but they all reach a saturation. When we still focused more on the controls, we were looking into the glucose metabolism. And already at the 0 point, it was clear that the glycated hemoglobin is actually correlating in the blood with the levels of NAD+. When we took all the time points throughout the whole clinical trial, then there was a strong correlation with insulin resistance, serum insulin, S-peptide, and the glycated hemoglobin in the whole blood correlated with both NAD+ and NADH, suggesting that there are negative effects correlated with increase of NADs over the normal range. We looked then further also into detailed about weekly where the NAD levels are going and where this glucose metabolism is developing. And you can see that the levels of fasting glucose, the fasting insulin and the HOMA index they are all increasing significantly from the point where in the blood the level of NAD+ is close to 50 micromolar NAD. Same here, the insulin is then significantly starting to increase from this much onwards. And we are not the only ones who actually show there is a negative effect of also increasing the NADs. And this is a study from Bergen University where they treated the Parkinson's patients with high doses of NR. They used 3 grams of NR per day. And also in this study, they showed that when they treated them with NR, then already from the 3rd day, there's a huge increase. There's an increase in homocysteine. There's an increase from the placebo control group to the NR study group. And the same as in our study, the increase when is from when the NADs are reaching close to 50 micromolar NAD+ in the blood. And then there's another study that came also very recently that they showed that an excess of NAD boosters or the excess of NAD and their breakdown points to 2-PY and 4-PY are actually having a vascular inflammation effect, and they are contributing to the risk of cardiovascular disease. So in a summary, our technology actually is the only technology that now can, from one sample, measure all the 4 forms of NADs and 2 forms of glutathione, including a blood sample. And we have verified that we can identify the patients that they actually are in the need of NAD supplementation. And then we also show that there is a negative effects associated with highly increasing NAD levels. So it's highly important to measure the NADs in the blood to identify the people, to be able to supplement, giving the guidance on the supplementation and adjust the dose according to the need of the patient. And here I would like to thank you to my colleagues, Lilia Euro and Professor Anu Suomalainen's group, and thank you for your attention.
Yeah, thank you, Jana. It's not often we hear about the negative effect of having too much NAD, and so I was delighted to have you tell us all about that. So would you think then that the decline in NAD that we see with age is actually an adaptation? It's a compensation for anything, do you think?
I actually don't think there is much of a decline with age, such as if we take a whole blood, there is actually a very strong decline in age.
Okay.
So there's publications, of course, of going decline in plasma, but in the whole blood we actually don't see that strong effect of decline with age.
Got it. All right. Questions? Anyone? All right, in the center here.
Thank you, Jana. Great presentation. So this sounds like a huge red flag for average members of the public who are just kind of randomly buying and consuming NAD plus supplements. Is that correct?
I would say so. I think it's very important to establish for every single person to know what your levels are, regardless of if it's using our technology or whatever technology. But I think it's very important to know. And I would strongly not recommend to go over the top, so going over to the saturation level, because I think, at least I have not seen any longitudinal studies of when people would take NAD boosters in high doses over years. Like, what kind of effects does it have if you take it for a year, 2, 5, 10? Mm-hmm. So I would really like to see this kind of studies before I would recommend somebody to take NAD supplements in high doses.
All right, thank you. Oh, is there a question in the middle? Yeah, there we go, behind.
Are these assays readily available for commercial use anywhere yet?
Yes.
Since it's so important, yeah.
Yeah, yes, definitely. We are already shipping we have the technology boxed in a nice kit box, so we can deliver them basically around the world, and we do that already. So you can just order it through our site, and we can definitely do that. We also have a service, so we can receive any sample types in our Helsinki laboratory, and we can analyze, we can collaborate. Everything is a possibility.
All right, thank you, Jana.